This is a production of Cornell University.
Perfect. Thank you very much. So thank you very much for being here today. I am Dr. Daniela Vergara. I'm the emerging crop specialist, which, as Mary was saying, it's a new position at the Cornell Cooperative Extension. I've been a cannabis researcher for a while. This is my email, my website, if you want to contact me. And I am going to be talking about my experience. So what I did, mostly research at the University of Colorado in Boulder before joining the Cornell Cooperative Extension. I am also the co-founder of the Cannabis Genomic Research Initiative, which is our group at CU Boulder, and the founder and director of the Agricultural Genomics Foundation, that it's a 501c3 nonprofit that aims to bring cannabis scientific findings to a broad public. And so I'm going to be talking about the things that I did, the things that I'm doing, and the things that I would like to do in the future. So for those of you who were at this meeting, it's a meetup that I am co-organizing with some people in the industry here in New York State. This is happening the second Thursday of every month. For those of you who were part of that, I gave a very similar presentation that day. So I am sorry if you were there as well. So I graduated from my PhD in 2013 at Indiana University, Bloomington, where I studied host and parasite coevolution. I was thinking a lot about why would individuals reproduce sexually instead of cloning themselves. And one of the reasons why you would like sexual reproduction instead of clonal reproduction would be to avoid disease and parasitism, particularly when you have a very taxing disease. So I was thinking about how host and parasites evolved. My degree is in evolutionary biology. And this is my advisor, Kurt Lively, at Indiana University in Bloomington. And so when I finished my PhD in 2013, I moved to Colorado. I was going to work with Nolan Kane, so he is my postdoc advisor, and he was coming from a different side of evolutionary biology, thinking about genomics. So genomics is the entire conglomerate of DNA, of genetic material of organisms, and he was working with sunflowers. So that's what he did in sunflowers, was look at the entire genome of sunflowers. So I joined his lab, and I was going to work with sunflowers, but around that same time they were legalizing cannabis, so marijuana recreational use, in 2014. So I started working in cannabis instead, and one of the things that really caught my attention was that cannabis reproduces both sexually and asexually, and I was thinking about those things through my PhD, so clonal reproduction. And so cannabis has hermaphrodites and has individuals that are males and females. So it has dioecious and monoecious individuals. So that was really what caught my attention when I started working in cannabis back in 2013. And again, cannabis is part of the family Cannabiaceae, along with hops. So hops is its closest existing species. They diverged approximately 25 million years ago. And so they are part of the same family. And again, hops is one of the main ingredients of beer. And they are angiosperms, so they are plants that produce flowers. And what I did mostly was study the genome, so study the DNA and the genes in cannabis, so the whole conglomerate of DNA, of letters of the DNA in an organism. So by definition, what I was doing was working with big data and mostly bioinformatics. And one of the reasons why I was interested in this genomics is because you can draw parallels to how the plant looks. So you can draw parallels between the genotype, the genetic material, and the phenotype, the physical characteristics. So the genotype, the DNA, and the phenotype, the physical characteristics of the plant. And one of the things that I did was to look at the genes that are related to cannabinoid production. There are multiple cannabinoids here. I'm showing two, which are of great importance because they are the drivers of this multi-billion-dollar industry. However, there's many cannabinoids, and I also worked with some of them as well. And I was part of a lot of different publications that for my research while I was there at CU Boulder. In 2017, for example, we published this paper where we looked at the cannabinoids, the 6 different cannabinoids from the cannabis produced by the federal government. So I'm not sure if some of you maybe know that in order for research to be conducted in cannabis, because it's a Schedule I drug, we must use the cannabis produced by the federal government, by the National Institute on Drug Abuse, NIDA. And so we were able to get a hold of the values, the chemotypes for different cannabinoids produced by NIDA. And I want to show you a picture. So we got a hold of cannabinoids values for the commercial marijuana that is produced by the private markets and by the government. And this is how it looks. So in this graph, what I'm showing in the y-axis, which is the vertical axis, is the percent cannabinoids between different locations. We had 4 different locations for the private market: Denver, Oakland, Sacramento, and Seattle. And we had NIDA, the National Institute on Drug Abuse. And NIDA differs in CBD, and THC is the one that has the lowest values. And when you look at the entire cannabinoid production, with this other statistical test, which is called the principal component analysis, that allows us to look at the entire cannabinoid diversity, we can see that this diversity can be partitioned into groups, this pink group and this gray group. And NIDA is only part of one of the groups. And then when we look at this diversity by location, Oakland and Sacramento, they contain NIDA's kind of cannabinoid variation, but NIDA does not contain Oakland and Sacramento variation. Then in 2021, so last year, we got another paper published regarding the genome of these NIDA's varieties. So I like to think about it like in 2017, we looked at the phenotype of the physical characteristics, and then in 2021, we looked at the genotype. So at the genome of these varieties, which were very, I'm not gonna go into details, but it was very limited, the amount of genetic diversity. So this is very important because most medical studies are using the governmentally produced cannabis. And this review paper in 2018 found that cannabis is only useful for 3 different conditions: pain in adults, chemotherapy-induced nausea, and spasticity in multiple sclerosis. And perhaps they only found that cannabis is useful for 3 conditions because they are using a source that has limited diversity, both in the physical characteristics, in the cannabinoids, and in the genome. So they're not using the cannabis that people regularly use in private markets. And again, this is how the medical marijuana from private markets looks versus the one that is produced by the government. I also worked with hops a little bit. So I was part of the assembly of the chloroplast genome. So the chloroplast is this organ in the plant, really important organ for photosynthesis, and it has a genome of its own. And basically what we did is that we pieced, so we pieced that genome together. And this was actually my first paper in cannabis in 2015. And I also work with hops and cannabis while describing the cannabinoid genes. I'm not going to go again into detail, but I'm happy to talk to you about it if you are curious about it. But part of this research while looking at the cannabinoid genes in cannabis, and I used similar genes in hops, this was published in 2019. I was part of different private companies, Steep Hill, which is a testing facility, and they have testing facilities all over the US. So I was part of their team. And then I was part of the Front Range Biosciences team. So these companies would fund me found my research at CU Boulder. With data provided by Steve Hill, we published this paper from NIDA that I just showed you. And then in 2020, we had another statistical analysis where we used machine learning to look at different cannabinoids and the missing values of different cannabinoids. And also in 2021, I was part of this study where we looked at the different physical characteristics of traits that are important for the industry. I am going to be talking about this particular paper on this seminars that are co-organized between USDA and Cornell, and my talk is on 420, so I hope that I see you there as well. And I also started a nonprofit organization, the Agricultural Genomics Foundation, that aims in bringing cannabis knowledge to a broad public. Through AGF, we have a podcast, Cannabis Science Today. I highly recommend it. And we also have a class, Modern Cannabis Science, that is taught at CU Boulder, and it's open to the public because it's taught through continuing education. So I made a lot of professional relationships during my time in Colorado, and personal relationships as well. And then I moved here to New York in October. I started this position at the Cornell Cooperative Extension. I am starting to get to know a bunch of people in the industry, which is really exciting. And as time passes, I hope that this slide gets busier and busier with multiple other companies. One of my tasks right now is to work on an introduction, a manual, a hemp production manual. And this has been a very collaborative work. In the survey that I'm going to put a link, or Mary or Janice are going to put a link in the chat. We hope to have people join us with this endeavor. So if you want to be part of it, please give us our email. Give us your email in the survey. And my idea is to build a cannabis community here in New York State and to establish myself as kind of the middleman between the industry and researchers, and to develop research and outreach programs that are going to serve the community and help this nascent industry thrive. Again, all of this is a collaborative endeavor with the manual and also with other things that we're going to be developing in the future. And I would like to be part of building this industry and answer many questions together. I think that there will be room for multiple businesses within the industry. Not everyone has to be a grower or a producer, and there will be plenty of room for ancillary businesses. And we're all learning how to process cannabis together. So that's something that we are in this together. And again, this is my email, my website. And with that, I will answer questions if you have any questions. Thank you.
All right, today we're going to talk about hemp micropropagation. Originally, this was supposed to be a live how-to demonstration. However, due to the COVID-related issues, this is going to be a PowerPoint presentation. So let me begin. I'm stationed at Cornell University's satellite location, the Long Island Horticultural Research and Extension Center in Riverhead. This year, we are celebrating our 100th anniversary, and I want to invite you down for Plant Science Day on September 15th. Before I begin to speak about hemp micropropagation, I want to take a few seconds just to tell you a little bit about our center. We are located in the heart of Suffolk County's agricultural industry. We are the only horticultural research center in the United States that has professional scientists who represent each of the horticultural commodities: floriculture and greenhouses, fruits and viticulture, vegetables, and woody ornamentals and landscape plants. In addition, we have specialists for the cross disciplines too. We have 2 plant pathologists, a weed scientist, and 2 entomologists. I represent the discipline of plant tissue culture that we're going to discuss today. Our research center is located down at the east end of Long Island. It is a little more than 300 miles from Ithaca. It's a long drive, but if you get a chance to visit, you will be surprised at the beauty of eastern Long Island and the amount of plant agriculture. Due to our unique maritime climate, good soils, high sunlight, and other environmental factors, we are still the #1 county for greenhouses in New York, as well as ornamental nurseries. We have more than 2,000 acres of grapes and grow more than 100 different kinds of vegetables. Okay, that's the end of my short little commercial for our anniversary. We're here today to discuss about plant tissue culture, and more specifically, plant micropropagation. Plant tissue culture is the growth of plants on a sterile nutrient medium in vitro under controlled environmental conditions. There are several uses for plant tissue culture. It can be used for plant breeding, disease elimination, basic research, nutritional studies, and much more. Today, we are focusing on the applied technique of micropropagation. Plant micropropagation is one use for plant tissue culture. This technique is used specifically for the clonal propagation of plants in vitro. This is a photo of our lab on Long Island. We also have a micropropagation lab on campus in Ithaca. Basically, you need to have a location to prepare media, autoclave it, laminar airflow hoods to transfer plants, and growth chambers or light racks to grow plants. Everything that you see here, you would also see in a commercial laboratory. Why is there such interest in using plant micropropagation for hemp? The main reason that micropropagation is popular is that it is a rapid procedure to asexually propagate clones. Because the female plants of hemp are the most valuable, it is desirable to propagate all female plants. The plants that are produced by micropropagation are clean and disease-free. With new virus problems popping up with hemp, it is nice to have a clean source of plants that grow vigorously. In addition, this technique does not require a large amount of space to produce the plants. Micropropagation requires less maintenance than plants that are grown in a greenhouse or field, and year-round production is possible. For the past 2.5 years, our lab has been working on the micropropagation of hemp. We have been studying the various factors that influence the growth of this plant in vitro. The medium on which the plants grow has several reasons to succeed or fail, so each aspect needed to be evaluated. We examined the plant's nutrition, amount of sugar for optimum growth, gelling agents for the medium, and the hormones that allowed the plants to grow most vigorously. In addition to the growing medium, we also examined other environmental factors on hemp. We looked at the type of explant that was used, the temperature to grow the plants, and the medium pH levels. I will tell you what the key items for success are, the ones that we found as we go through this presentation. There are 5 stages of micropropagations, and I will review each one of these in detail. First, there is stage O, stock plant selection and preparation. Stage 1 is the disinfection and introduction of plant material into tissue culture. Stage 2 is the multiplication stage. Stage 3 is rooting, and stage 4 is acclimation out of the culture vessel. Okay, I'm going to give you a spoiler alert now. We found at the end of this research that stages 3 and 4 can be combined for hemp, and this shortens the process, decreases costs, and increases efficiency. Stage O is for stock plant selection and preparation. Remembering that micropropagation allows you to produce hundreds or thousands of plants, it is critical that you begin with the correct cultivar. It is best to begin with plants that are healthy, well-fertilized, and disease-free. Results of the plant's growth can sometimes be affected by the time of year when the plants are placed in vitro, the day length in which the plants are growing, the light intensity, and several other environmental factors can affect the plant growth. During our research, we examined the effects of where the stock plants were grown on the establishment in vitro. Generally, explants that are taken from plants that are grown in the field are the most difficult to establish in vitro. Explants that are taken from plants that are grown in a greenhouse have a better chance, and those that are taken from plants that are grown in a growth chamber are best. By removing plant material from plants that are growing in a clean environment, you increase your chance of success at stage 1. Stage 1 is the initiation and establishment in vitro phase. Usually a simple diluted bleach solution is used to clean plant material. We tested various levels of Clorox bleach for 10 minutes to see how well the plants were cleaned. You can see some of our results here, and there are 2 important takeaway observations. First, explants that were removed from plants that were grown in the growth chamber had a much higher success rate than explants that were taken from plants that were grown in the greenhouse. Second, the 20%, 40%, and 60% bleach treatments All had excellent cleaning abilities on the explants that were taken from the growth chamber. Any of these levels work fine and there's no damage to the plant material. Based on these findings, we recommend 20% bleach for 10 minutes to clean hemp explants for culture. Other ways to improve your chances of obtaining a clean plant during stage 1 is to use new shoots when possible. They are cleaner from fungi and bacteria than older shoots. Remove your explants from young plants. They are better to respond than older plants. Juvenile plants work better than adult plants. One of the things about stage 1 is that if it wasn't successful, you will visibly see contamination within 3 to 5 days. Here on the screen, you see different examples of fungal and bacterial attack after unsuccessful stage 1. The objective of stage 2 is the rapid multiplication or propagation of propagules. During stage 2, the plant material from stage 1 is repeatedly subcultured until the desired number of plants are obtained. A key part of the success of stage 2 is the choice of cytokinins and auxins, as well as the concentrations of each that are used. This is a photo of 5 different media that were used to grow the same African violet plants. Media that had cytokinins and low levels of auxins are on the left. As you move to the right, the levels of cytokinin decrease and the levels of auxins increase. You can see how the hormone level in the medium can affect the growth of the plant. More shoots are obtained with higher levels of cytokinin, and roots or callus can be produced when cytokinin is absent and auxin levels are high. During stage 2, there are 4 techniques that can be used to micropropagate plants. However, there is only one that can be guaranteed to produce genetically identical clones, and that is number A on this list. Use organized growing points such as shoot tips and axillary buds. There are other ways to generate plants during stage 2, such as somatic embryogenesis, adventitious shoot production, and seed propagation. But none of these techniques can guarantee clones, so I will not discuss them today. We found that hemp plants usually grow in vitro by growing a single shoot with no or very little axillary branching. This is an acceptable way to micropropagate these plants. Plants such as lilacs, rhododendrons, mountain laurels, and others will grow in vitro by producing multiple axillary branches. Hemp does not respond this way. As I mentioned earlier, during our stage 2 research, we focused on several different media factors for optimal growth. We looked at sugar levels. And by the way, for plant tissue culture, you can use plain old table sugar. We compared levels from no sugar at all the way up to 60 grams per liter. Usually 30 grams per liter is the standard. We found that either 15 grams per liter or 30 grams per liter were the best levels to grow hemp. We chose to use 15 grams per liter because it saves money and you get the same results. We also compared 4 different commercial media formulations: Murashige and Skoog, indicated by MS, Linsmeyer and Skoog, LS, Driver and Kuniyuki walnut media, DKW, as well as Lloyd and McCown's woody plant medium, the WPM. All the first 3 media performed equally well. The only medium that had poor plant growth was the WPM medium. MS medium is the industry standard around the world, and we recommend that medium because of convenience. We also compared gelling agents in the micropropagation medium. We compared agar, which is the most commonly used gelling agent, to gellan gum, which is a water-soluble polysaccharide. Commercially, it's called Gelrite. And then we also compared these to an agar-gellan combination. There were no significant differences between these 3 gelling agents. Any can be used at their recommended concentrations. We also compared the explant source for stage 2. We compared shoot tips to nodal sections. There were no differences between shoot tips and nodes. Either can be used equally well. This is a good thing for commercial propagators. Another batch of experiments focused on the pH levels of the medium for micropropagation of hemp. We tested pH levels of 4, 5, 5.8, which is the industry standard, 6, and 7. We found that the best pH levels were 5.8, 6, and 7. We recommend that the standard pH level of 5.8 be used for hemp. The optimum growing temperature for hemp was also not known. We grew hemp plants in 4 different growing temperatures. We went from as low as 22 degrees Celsius, also 72 degrees Fahrenheit, all the way up to 28 degrees Celsius, about 82 degrees Fahrenheit. The two warmest temperatures, 26 degrees and 28 degrees, had the best plant growth. These are recommended for hemp micropropagation. A major set of experiments examined different possible cytokinins and different levels of each. We compared benzoaminopurin, BA, to isopentyl adenine, 2IP, as well as to thidiaziron, which is indicated by TDZ. This is not a cytokinin, but it is a chemical that has cytokinin-like activities. Each were compared at 1, 5, or 10 micromolar levels. The lower rates of 2IP or BA performed the best. These growth regulators produced more shoots than the control plants that had no growth regulators. These two cytokinins are what we recommend for hemp at the lower concentrations. TDZ produced cultures with greater number of nodes, but the shoots were short and callus was generated. Thidiaziron is not recommended because it produces callus, and mutations are highly likely when plants are produced indirectly through callus. The last two stages of micropropagation that we studied were stage 3 and stage 4. Stage 3 is used in traditional micropropagation for root initiation. Stage 4 is used to harden off or acclimate plants. Stage 3 for root initiation is to transfer the hemp plants to a new medium with either no cytokinin or to a medium that includes an auxin. We compared the two most commonly used auxins, NAA, which is naphthalene acetic acid, and IBA, indole-3-butyric acid. We compared them at 4 different levels. We had a control with no auxin, 0.25 micromole, 0.5 micromole, and 2.5 micromole of each. We are recommending 2.5 micromole IBA for rooting hemp. But as you can see here, roots are also possible with NAA. There was a lot of callus produced when NAA was the auxin, and because we do not want to encourage callus, we think that IBA is the way to go. Stage 4 is the acclimation or hardening off of micropropagated plants. Tissue culture plants are grown in 100% relative humidity, so the plants do not form cuticle wax on their leaves. They cannot be removed from tissue culture and placed outside in the greenhouse or field. They must be hardened off. This humidity surrounding the plant needs to be gradually decreased. Because the process to generate a waxy cuticle on a leaf will only take 24 hours under good conditions, you can simply crack open the culture vessel overnight. Most often, however, plants are removed from culture and transplanted into greenhouse media. They are then placed in a high humidity environment and then has the humidity lowered gradually. Another thing to do to micropropagated plants when they are removed from tissue culture is to gradually increase light intensity and photoperiod. Plants in tissue culture do not photosynthesize well because they have a sugar source in the medium. By increasing the light levels and also giving them long days, the plants will begin to photosynthesize and grow better. Some stage 4 experiments compared acclimating and rooting hemp plants under intermittent mist or under a plastic dome. We also applied rooting hormone to some of the plants and no rooting hormone to the other half to determine if rooting was improved by the application of auxin. The plants on the right side of the slide were rooted and acclimated under a plastic dome, and the plants on the left side were placed under greenhouse intermittent mist. Plants that were grown under plastic domes rooted faster, and the plants were healthier than plants that were placed under the mist. The application of rooting hormone did not have any advantage to the rooting process. We found that rooting and hardening off of micropropagated plants can easily be done together at stage 4. For hemp plants, there is no need to go through a stage 3. Time and money can be saved by combining these two stages. Okay, that's everything you need to know about micropropagation of hemp in just 20 minutes. I appreciate your attention. I hope this was helpful. I want to thank the young people in this photo. They are the members of both our Ithaca lab and our Long Island lab. They do the real work for this research. Thanks again, and I can answer some questions now, if there are any.
Okay, I'm going to have one, maybe was everybody else. So you're talking about callusing. Why is that bad? What is it?
Okay, callus is just a group of unorganized cells, parenchyma cells, basically. And when a plant is regenerated from a Cala cell, there is a much greater chance that there's a mutation that occurs. And we want, for this situation, we want clones. So if we want clones, we need to start off with organized growing point, which are basically the shoot tip or those shoots in the axillary buds.
Okay, cool. Which is why you don't want it early on, because then you're going to take that first plant and make other little copies from that one. And so you don't want callus form on that first.
That's right.
So that, okay. Yeah, I get it.
Well, thanks a lot, Mary. Thanks for the invitation. Good afternoon, everybody. I'd like to actually jump in on your comment there, Mary, that even though I've been looking fairly intensively at some of these, particularly foliar diseases on hemp over the last several years, it's still awful nice to have a microscope back at the lab and the ability to grow these things out, because there's just enough difference between cultivars, the genetics of hemp and the environment, location, that you can still get pretty easily fooled. So I don't want to start out making you all think that you can just take a look and know what it is by comparing it to some picture book. But let me share some of the things we've been finding. And for starters, I want to acknowledge, when I started into this work, there is not a heck of a lot out there, or wasn't at the time, and the literature is building nicely now. But I want to say hats off to J.M. McPartland, who is actually a physician who was interested in some of the medical psychoactive portions of the hemp plant. And he put out a beautiful textbook at the time on hemp diseases and pests. And that's been a great starting point for learning about a lot of these things. So let's start with some of the ones that are kind of universal, that if you grow hemp for much time at all, you're probably going to run into white mold. And this is caused by the fungus Sclerotinia sclerotiorum. It's the fungus with a very wide host range and is problematic in New York on a number of our vegetable crops. One of our biggest problems on soybean production. So this is soil-borne. The overwintering stage of the fungus is these hardened bodies called sclerotia that reside on the soil surface or within the soil. And white mold results from that, especially if we get the little mushroom fruiting body formed in the field about the time that hemp is starting to flower. We can get infection. And I think one thing I want to call your attention to here is the concern about your crop rotations, what other crops you're going to grow in rotation with hemp. And you want to be particularly cautious if you're in your rotation with a number of particularly legume crops. They can be vegetables, they can be things like soybean. Sunflower is a very susceptible crop, but anyhow, be cautious of what's in the crop preceding hemp and what diseases have built up there. Gray mold is probably the most universal thing that I can almost guarantee anybody growing hemp, whether it be in the field or in a controlled climate situation, you're going to run into this gray mold, botrytis, at the end of the season, and it can cause problems because it grows all over the buds, the flower parts, and in fact other parts of the plant, and can cause a seed quality, a bud quality issue in the eventual use of this crop. Again, it has a very wide host range. You're probably not going to avoid this by rotation, but something to be aware of generally. Powdery mildew, I won't say much about it because Chris Smart and her group have worked on this, but it's something you're likely to see at least low levels of in the field. And if you're in a controlled climate, humid situation like a greenhouse, you're very likely to run into more of this. So I won't steal Chris's group's thunder, and they will be talking about this. I think all of you know that the early part of the life cycle of a hemp plant, when you plant seed or seedlings for that matter, the crop is not really competitive at its early stages. And amongst the various things that plague it are the oomycete problem, Pythium species causing Pythium wilts. And compared to a number of other vegetable ornamental crops, hemp is particularly susceptible. And we see this in both field and greenhouse operations. And we've worked for a couple of years now with Alan Taylor at the Cornell Agritech. And Alan's worked out a nice, if we can't always get out and look at this in the field setting, we've got some nice assays there where you take infested field soil and you bring it back into the lab and dilute that out with sand and use these ragdoll systems where you germinate the seeds. And what we particularly see is in post-emergent seedling death. And this is 4, 7, 10 days after planting these seeds in these systems. And we've been looking at this with Alan to hopefully identify some seed treatments, both chemical and biological, that can help get around this. Unfortunately, at this particular point, none of the approved available biological treatments are really doing a very significant job. Some of the chemical fungicide treatments are doing well, but there's next to nothing labeled for hemp. So that's a continuing challenge. And when we've looked at pathogens coming from soil in this assay that Alan's developed, it's not only Pythium, but there are a number of other soil-borne fungal genera, species such as Fusarium and Rhizoctonia in particular, can also give rise to the damping off symptoms. So the Fusarium fungus is very cosmopolitan. We find it in many soils, and certainly we have seen instances where Fusarium, particularly Fusarium oxysporum types, can infect, usually through the roots or crown of the plant. And depending on the timing, the growth stage of the plant, we can see premature death of plants. We can see crown and foot rots or complete vascular wilt that kills off the plant. I'll talk a little bit also about certain Fusarium fungi can infect through the flower parts and cause a flower or bud blight. And we're concerned about that, not only because of the damage it does directly, but the Fusarium fungi produce a number of mycotoxins that can be harmful to humans and animals. So that's an issue downstream in the products. So we have found several species of Fusarium associated with these flower blights, but predominant amongst them, we find Fusarium graminearum, which I have spent the better part of my career working on in grain crops. It's the cause of, our principal cause of ear rot in corn and of Fusarium head blight in things like wheat and barley. And there are a number of mycotoxins, but principally we find the toxin called in the vernacular vomit toxin because of the effect that it has on animals, but it's known as deoxynivalenol, or shortened to DON. And in grain crops, we are concerned at levels above 5 parts per million. And for human food, you don't want more than 1 part per million. That's milligrams per kilogram of body weight in a total feed for a human or an animal. So we have indeed found levels in harvested hemp grain well exceeding 5 parts per million. We did some brief experiments, preliminary experiments, where we pressed with a commercial oil plant. We had some of that grain pressed to make oil, and most of that resided in the press cake that's left behind, and relatively little DON went into the oil. So that's one piece of good news. Increasingly, there's interest in what the Don toxin will, how important that will be as a contaminant in the CBD product chain. So I don't mean anybody to read this table. On the left-hand side, there's a list of very commonly occurring fusarium species that we see here in New York, particularly on grain crops. And each column across that are unique types of mycotoxins that are produced. So what I'm saying is we're keyed in at this point on one of those toxins, deoxynivalenol, but we find some of these other fusarium species. So more work really needs to be done to see if any of these other toxins are potential issues in the processing of products for coming from hemp. So in the first couple of years that I worked with hemp in New York State, we did survey work. I found only 1 or 2 fields with Septoria leaf spot, also known as yellow leaf spot. Now I would say that this is our most prevalent foliar disease in New York State. These spores are born in the residue of old crops of hemp. I cannot rule out that they might not be surviving on other crops. We just don't know enough. But this Septoria cannabis, you'll start to see these lesions on the lower part of the plant canopy as the season progresses. With splashing rain, the spores are splashed increasingly upward in the canopy. The spots expand. They have pronounced yellowish margins around the brownish dead areas. And we have seen these now almost becoming more prominent each year in the last 3 years. And we saw quite a lot of this in 2022 as well. So I have a little video here of disease, or kind of what it looks like in a canopy. These were a little movie put together by Jen Starr. And you can get down in the canopy and see a little bit what the development of this Septoria leaf spot looks like. And again, for a lot of these things, you look first on the lower leaves, and as the crop progresses, you'll see these things under the right environment move up in the canopy. So the first couple of years that we started working on hemp in New York, the most prevalent thing I saw was bipolaris leaf spot. And the folks in Kentucky were the first to characterize this. They refer it to as hemp leaf spot. I kind of don't prefer that because all of these are hemp leaf spots. So I like the name that with the organism that kind of distinguishes this. But these start as kind of pinpoint white spots on the leaves, scattered specks. And as they grow and develop, you can see in some of those pictures there, they have kind of a darker edge to them with kind of a light tan or whitish center. Now, as those grow and coalesce, that pattern may go away and they can become looking a lot more like the Septoria, for instance. So that's just why these things are kind of complicated, and you almost need to look at your field, scout them progressively through the season. This is an interesting one that we need to learn a lot more about. It's a documented pathogen of number of monocots of grasses and also of some dicots and weeds, things like velvet leaf. We don't precisely know the epidemiology in a place like New York with this yet, but this is probably the most frequently reported foliar disease across everywhere that hemp is grown in North America. So I've got a little close-up of this as well in the canopy. And you can see how it kind of has that kind of bleached look before it starts turning darker. Sets it apart, at least from the early stages of the Septoria. So a couple of years ago, we first discovered in some of the Cornell plots in Ithaca the presence of an oomycete pathogen, downy mildew, and this is an obligate parasite, so it survives in green living tissue. what you see here, you really need to look at both leaf surfaces here. On the upper leaf surface, you see this kind of non-distinct light green lesion, but you turn that over and you see these darker colorations, and you see the the sporulation of the causal oomycete under humid gray conditions, like when you still have dew out in the morning is a good time to look for this. And we have seen this now for a couple of seasons under fairly, in a couple of different locations, but at this point, at least not widespread across the state. And these are the characteristics, spores, the sporangia and the sporangiophores here that you may see under a powerful hand lens or under a microscope. And a little video of this. As these lesions progress, even on the upper side, they become more angular and much darker in color. As you can see, they kind of fill in those leaf panels in kind of a very sort of angular shape. Very distinctive. And we don't know enough about this yet, but I think working with Larry's program and Chris's program in the future, they'll be looking at different varieties, different cultivars. And just a quick look at this, we looked at 3 different ratings on the hemp essential oil cultivars, and you can see there's not a complete resistance, but there are definitely some cultivars by comparison that show less or slower development of downy mildew. So one really striking finding a couple of years ago in 2019, in the variety, the cultivar plots located in Ithaca, New York, we found a foliar rust, which is really striking because this had not been described in any way in North America. And we found these, the yellow orangish pustules, the orange rust stage of this rust fungus, and almost exclusively on the lower leaf surface. So you could easily miss this going through a field. You might see some light yellow spots on the upper surface. You really need to look on the lower leaf surface. We have seen this now recurrence in all 3 of the last growing seasons and in a few different locations, but still not widely dispersed across the state. And this is also have been observed in sort of the Mid-South region in Virginia and Tennessee. This is what the Uridinea spores, those orange rust spores, look like under a microscope. And again, these were born on the lower leaf surface. So the book I showed you initially referenced this as an issue in a few places in Northern Europe. And it was originally described by a person called Krieger, a mycologist, and it was identified as Uredo, which is kind of a bucket of taxonomically of related genera of rust fungi. It's not a finely defined genus, but he described this as Uredo kriegeriana, and we were extremely lucky to find part of a type specimen in Cornell's herbarium, and this dates to more than 100 years ago, found in Germany. And you know, there's been no recent citation in the literature of rust as an issue, frankly, anywhere in the world, even in Northern Europe. So we tried to get DNA from the limited sample that we had here to try to compare sequence with the modern one, but we weren't able to get DNA out of this ancient sample. But we were able to compare the description of the spores, matched up very well with our modern sample. And we have now deposited sequence for the isolate from New York. Over time, we have seen a lot of just a few other things that we see off and on, Cercospora, we see Stenophyllium leaf spots, but none of these have really become that common across the state. We may see them with one cultivar in one location in 1 year. And a number of other pathogens such as Calatatricum species, et cetera. So this is kind of a continuing, evolving scene. One thing that fascinates me very much is what's the life cycle of these things in terms of the host range. So for instance, we saw in the same field that we saw Cercospora on the hemp, we saw a lot of Cercospora on lamb's quarters weeds in the field. They may or may not be the same, but I think a lot of these kind of puzzles need to be solved. This is one that we have seen a couple of years ago, but in the very wet growing season of 2021, we saw a number of fields where this was far and away the most extensive blight. And this is a fungus called Cristulariella or Growsinia. And we found this under the very wet conditions, very prevalent across many varieties, even across CBD varieties and forage type varieties. And here's an interesting thing. This fungus has a very wide host range. And we noticed in the one location in the southern tier of New York, we saw a box elder tree that was just loaded with these zonate leaf spots. We collected the fungus from the box elder as well as from the hemp. We sequenced DNA and we got 100% identical sequence to the Growsinia moricula match with both our hemp and our tree isolate. So that's fascinating. And again, comes back to the question, what is the host range of some of these things that we're finding? I won't talk about this, but occasionally we see a bacterial leaf spot. This is one discovered in the SMART lab. This is a red pigmented bacterium, Serratia marcescens, that can also be found as a blight. So very briefly, I think many of you know this already. If you go to the NISPAD site of New York Department of Environmental Conservation, look at registered pesticides for New York. If you initiate a search there and go into the next level where you do an advanced search, and then down on the bottom left there, you click industrial hemp, it will show about 107 different products. These are by and large least toxic chemicals, biological treatments, things that are kind of generally available for organic control. Many of them are on the OMRI list, but this is a source of seeing what things might be available. We don't have, I know that Chris is going to talk about this in a few minutes here. We have done very limited look at some of these necrotrophic leaf spot pathogens and have not seen anything really overwhelming control with any of these materials, but some people have looked at some of the biological control materials and reported, at least anecdotally, some positive results. So what do you do in the midst of all this? First of all, the vast majority of these diseases are not directly hurting yield and quality. So really key in on those that develop early and actually are attacking either the flower part of the plant or attacking the foliage early enough to reduce yield. Prevention's our best tool still. Some of these things are seed-borne potentially or soil-borne. So paying attention to getting clean seed, having good soil health practices, spacing out your plantings to reduce humidity and get airflow in the canopy. A really good idea to remove any debris of hemp, of a previous hemp crop off of your field and either burn it or completely compost it. Weed control is important. I mentioned a little bit of the part we know. There's a lot we don't know in terms of crop rotations, where white mold is an issue. in some of the legume crops or other vegetable crops, you want to be cautious there. And in terms of mycotoxins, the fusarium mycotoxins on the flower parts, probably corn and small grains rotations are not a really great idea, which is tough because for other reasons they'd be excellent rotational crops. So there's a lot we need to know yet. And in addition, we talked about things that could be sprayed. I think a lot of emphasis in the future will be on looking at cultivars that are more resistant than others. So in terms of diagnostics, I'm kind of a short termer in this business. I will not be entertaining diagnostic samples in my lab this coming growing season. Chris Smart may or may not be looking for certain type of samples, but I want to make you aware that the Plant Disease Diagnostic Clinic based in Ithaca is really geared up to look at hemp samples this year. Sandra Jensen has worked with us in the last 2 growing seasons to really become attuned to diagnosing hemp diseases. But this is, through the clinic, a fee-based service. So just some kind of pointers on sending plants in, particularly foliage samples, is the cardinal rule: don't add any water to the sample. Just flatten out your leaves and put them in dry paper towels. You can put them then in a Ziploc bag, but whatever you do, don't put moisture into that Ziploc. And these can be, the sooner they get to the lab, the better. Usually you want to avoid shipping them over the weekend. And it's always a good idea to call the lab that you say you have something coming. So with that, Mary, that's my comments. And if we have time for a couple of questions, I'd love to answer them.
Okay, we got one in chat. What are the best cultivars to use in New York? Well, we're going to have a lot of chat. Well, not a lot of chat, but there's other cultivar coming up. But I think maybe since it's Gary, are there any cultivars that you've seen that are particularly disease resistant?
Not really. I mean, for most of these things we've been talking about, if you look at septoria, look at downy mildew, you look at several things. You're looking at partial resistance would be my best guess. You're looking at shades of gray. We haven't purposefully done a lot of that work, other than we've tried to look at Larry's plots in Ithaca and Geneva and whatnot, and just look for whatever Mother Nature provided. We have not done any of what I would do in my cereal crops, for instance, is actually inoculate plots and, take no have no, take no prisoners. And so we can make some very general conclusions at this point, but maybe one of the later speakers, there's no miracle cultivar at this point. And we just don't know enough to make that kind of a broad representation. I would say, as somewhat of a generalization, the CBD type varieties seem to be quantitatively more susceptible as a class than the fiber or the grain types. And again, that's a little subjective because that's going out and looking at the variety trials, and that's not a direct comparison where it's been inoculated.
The only thing I'll throw into that is because the CBD types are typically grown more like tomatoes, very far away from one another, instead of like the grain types where in there it's like a jungle.
Yep.
I've never seen sclerotinia on CBD types, but in the grain and fiber types, it's not hard to find at all. That's just me, what I've seen. So that to me is about the environment you provide just based on density.
Well, that's a good point. And air movement in these canopies is critical if you want to tamp down some of these fungal diseases. They love moisture and humidity. I have seen sclerotinia in white mold in CBD types, and even in hoop houses where the soil that was brought in from somewhere to put in the hoop house had the sclerotinia in it. So It's not even just crop rotation in the field, but think about where your soil's coming from, what other crops it's been exposed to.
I do have a couple of just general comments or one comment, two questions. Pythium. This, I know, I can hear Chris laughing when I say it. This crop is shamelessly susceptible to Pythium. So I'm going to bring back, Mark's greenhouse, and we've got some other stuff going on that, like, sanitation,
Right.
all the time, everywhere, no matter what. Just throwing that out there. Other questions I have. So on the downy mildew,
Yeah.
clearly that's a different genus and species, which people on the call need to know that there's a lot of different genus and species that we call downy mildew across a variety of, crops that we grow. So that's one, they're not all the same downy. But two, is it as quickly, does it spread as quickly? Does it, take over a plant as quickly? I mean, downy on things like basil and some other crops, I mean, in a matter of days, it absorbs the whole thing. Yeah. And is really hard to control. Even if you have a fairly decent chemical for it, just forget it. If you don't get it the first 24 hours, it's done. There's no beating it back.
Well, the two parts to your question, the first one is that we did see, under favorable environment, fairly rapid progress. It's not like in a greenhouse setting, but yeah, it was fairly rapid progress. On the other hand, I don't have a good feel for spread at this point because we've seen them in very limited geographies. So, there's a lot to learn there, but it hasn't been a distributed problem so far, just in a few small locations. But you know how that goes. Once something spreads, then,
right.
Yeah, and we'll continue to discover new things. I like your point, Mary, about common names and even genus names. So for instance, when I talked about Septoria blight, well, there's Septoria blight on tomatoes, on wheat, on other, those are all different pathogens. Okay, so just because something has a Latin name in the title, that also does not mean it's not different use of that common name,
right.
in different crops. So that's a great point. And yet some of these things, just because of the way they're appearing and the patterning, I think probably have another host out there, whether it's a weed or a grown cultivated crop. So it's going to really take a while to ferret out some of that business. But
and my other question was, you mentioned the mycotoxins, and I assume we're talking about for seed production for either human or animal consumption. That's what we're worried about.
Yeah.
So are there standards for Canadian hemp seeds in terms of how much of what mycotoxins, so that, because they certainly have a pretty big market on that?
Well, for all human food products, it's basically 1 part per million. That's milligrams per kilogram. And that's for broad variety of commodity crops. So I would think this wouldn't be any different. And there, at least at one point in Canada, if it's something that goes into baby food, it's even more. It's like a 0.5 part per million. Not I don't know if
no, I was just
baby
curious,
food, but it's,
they're making most of the seed right now, or any of this.
We had a really critical situation with field corn in New York State this past growing season with DON in harvested corn. I mean, it's a really bad situation, and in some mills that are taking it for like ethanol, the average is coming in with 3 parts per million.
Wow. In
corn. And we have, I won't name names, but we had one place that had 50 parts per million in shelled corn this past growing season. So we definitely don't want to run into anything like that with hemp, okay? Because that's the sort of thing that just can kill a new crop. So my email is gcb3@cornell.edu. I'd welcome questions if they don't occur to you right now.
All right, so I'm Patrick McMullen, one of the graduate students in Chris Smart's lab. And today we're all going to be discussing results from our 2021 disease field trials. So the particular disease that I work primarily on is Fusarium vascular wilt of hemp. And as you can see from this image here, Fusarium vascular wilt causes a pretty severe collapse of the plant if it reaches these later stages. And we've seen this across New York State in a number of different scenarios. So first, I'd like to discuss some of the disease signs and symptoms that we most commonly witness, some of our current management strategies available, and then some results from our fungicide and growth promoting inoculation trials, and a new project that we're beginning with copper fungicide treatments. So first, Fusarium vascular wilt can be caused by a number of different Fusarium species. Primarily these three listed here, Fusarium oxysprum, solani, or proliferatum. And we've seen all three of these in New York State so far, with Fusarium oxysprum being the most common by far. Here are some pictures that I've taken in earlier field seasons, where if we start over here on the right, there's this young transplant that the root system never fully developed outside of its initial plug, and the roots were bound and unable to grow enough to support the plant. And then over here on the left is an image of an older, more mature plant that suffered a similar scenario where these roots were not large enough. There was not enough root biomass to support the upper parts of the plant, and the plant wilted and collapsed. And some of these characteristic symptoms of Fusarium wilt are some reddening of the roots. It might be a little hard to see in this image, but reduced root biomass and reddening of the roots are common. So a little bit more about the pathogen. It primarily infects the root system of the plant through these little tiny microconidia that can infiltrate through the root hairs, and these will germinate and produce hyphae and mycelium within the xylem, the water transporting vessels of the plant, where a mass of hyphae will accumulate and block water flow to the upper parts of the plant. And that's what leads to the wilting and plant collapse. So the disease cycle that we've seen in the field, it primarily occurs where we have these asexual spores, like we saw in the last slide, that can survive in the soil for a long time, and then they'll germinate, enter the root system, and as it colonizes the xylem, like we said, it will block water flow. And at this point, this is when you start to see the red or browning of the root tissue and the xylem, and then you'll start to see yellowing on the upper parts of the plant, and eventually wilting, chlorosis, and complete collapse. And when the plant collapses, that's when these new spores will be released into the soil. These hardy chlamydospores, they're called, can survive in the soil for quite a long time. So it's a difficult pathogen to eradicate once it's there. Some of our management strategies right now are quite limited. We don't know of any genetic resistance, which we often rely on genetic resistance as our first line of defense. But some good sanitation practices can help reduce disease in the field. So it's important to remove infected planting material. Gary mentioned this a bit in his presentations, that you have to be careful where you're getting your soil from. Make sure you're not moving soil between fields, especially in diseased fields. Ensure adequate drainage in the field, since this fungus likes a moist environment. Avoid planting in those low wet areas of your field. Crop rotation may help, but this pathogen does have quite a wide host range. And as we've seen with most diseases in hemp, approved fungicides are fairly limited and efficacy is often unknown. So to answer this last question, we set up a field experiment 2 summers ago, or last summer, that studied some commercially available products and their efficacy in controlling Fusarium wilt of hemp. So we tested a copper-based product from VM Agritech, and then some different biological strain products. So we had a Pseudomonas inoculant, Bacillus, and a Trichoderma product. And then we inoculated the field with Fusarium. And unfortunately, we just had low disease pressure over that season. Our inoculation method wasn't quite substantial enough that we could adequately evaluate these products. So these are some areas of research that we're still interested in trying to evaluate what products can effectively control Fusarium wilt. This pathogen can also cause damping off, like we've seen with Pythium and some other soil-borne pathogens. On the left here is a flat that was not inoculated with Fusarium. And then on the right here is one that was, and you could just see the significant reduction in germination. So I'm currently working on a product to see how we can prevent some of this damping off and what available management strategies we have. So I'm currently using a copper-based fungicide, a newly developing product from VM Agritech. And here I just plated the fungus Fusarium oxysporum on our Petri plates. And these have increasing concentrations of the copper fungicide from none, which is our control, all the way up to 200 parts per million. And you can see as we move across the gradient, there's less fungal growth. And then once we get up to 75% or 100% of these concentrations, the 200 parts per million or 150, there's very little growth, if any, of Fusarium. So now we're working with Alan Taylor's lab to determine a strategy to apply this product as a seed coat in hemp to see if this will increase germination rates. So essentially what I've seen so far is that I've been studying Fusarium oxysporum is a soil-borne pathogen, can survive in the soil for a long time, and is quite difficult to manage and eradicate. Management strategies at this moment rely primarily on cultural practices, since we don't have good fungicides available and we don't really know much about their efficacy. So more research is needed on the efficacy of these fungicides, and the research for the copper fungicide seed treatment is ongoing. And now I'll hand it over to Ali to talk about powdery mildew.
Great, thanks, Patrick. My name's Allie, and I'm also a PhD student in Chris's lab, and I am working on powdery mildew on hemp. If you've ever grown just about anything, you're probably somewhat familiar with some kind of powdery mildew, but in case you're not, this powdery mildew is caused by Golovinomyces ambrosiae, and it's characterized by asexual spores on the surface of the leaf, and it's also a pathogen that needs living tissue to grow. So we can't culture it. We need to grow it on whole plants when we work with it in the lab. And so these, the asexual spores on the surface of the leaf, you can see here on this first picture on the left. And then if you were to zoom in on that leaf, you would see those canidia in long chains, like you can see in this middle photo here. And then on the right, there's a photo of what those individual spores look like, and those spores are carried in the wind and can cause new infections on healthy tissue. So next slide, please, Patrick. Okay, so for this project, I was looking at ways to control powdery mildew, particularly looking at fungicide use. So currently, there's not too many options to control diseases with fungicides on hemp. What is labeled for hemp in New York State are mostly OMRI listed, and most of those are biologicals. And then more importantly, what is listed, there's limited publications on the efficacy of those fungicides against hemp pathogens specifically. So thank you, Patrick. So the goal of this project is twofold. So first, to compare some of those currently labeled products that are available to use on hemp for use to control hemp powdery mildew. And then the second part of this project was to look at whether both powdery mildew infection or fungicide or the fungicide treatments we were testing would affect cannabinoid production in hemp as well, since we know that a lot of folks are growing hemp for cannabinoid production. We thought this would be a good question to look at. Next, please, Patrick. So in 2020, we did our first fungicide trial with some of these products. And these are the products that we used in that trial. So we had Lifeguard and Double Nickel, which are two different bacillus treatments. And then we had a treatment where we were alternating the Lifeguard and the Double Nickel every other week. And then we had Silmatrix, which is a potassium silicate. And then lastly, we had azoxystrobin, which is a conventional fungicide. And the azoxystrobin, we expected to essentially work against the powdery mildew, since we know azoxystrobin works for other powdery mildews. But azoxystrobin is not currently labeled for use on hemp. So it's not actually something that a hemp grower could use, but we were using it as more of a positive control for this experiment. And then we also had an untreated control plot as well. And then we applied all of these weekly throughout the season, starting before our first inoculation. Next slide, please. So in 2020, this was how we set up our experiment. So we had, we used the cultivar TJ CBD, and we grew up our inoculum on full plants in a growth chamber. And then to make up an inoculum, I rinsed, washed infected leaves with a water and tween solution, and then sprayed that solution on plants using a backpack sprayer. And then I did this 3 different times. And then once we got disease in the field, I rated those plants 5 times, once a week. And I rated them for percent disease coverage, and then calculated the area under the disease progress curve. And then I also collected inflorescence or the flowers at the end of the season for cannabinoid analysis to address that second question. Next, please. Okay, so this is our results for that first trial in 2020. So on our x-axis here, you can see the different fungicide treatments. So we have our untreated control, Lifeguard, which is the first bacillus treatment, Silmatrix, which was that potassium silicate, Lifeguard alternating with Double Nickel being the second bacillus treatment, Double Nickel by itself, and our azoxystrobin. On the y-axis was the area under the disease progress curve, and those letters on top of the bars indicate 2 keys groups. So if the bars have the same letters, they're statistically the same to the same letters to each other. So you can see in this graph, the large bars indicate more disease. So our untreated control had the most disease, and then our azoxystrobin had the least amount and did a really good job at controlling the powdery. But like I said before, the azoxystrobin is not actually labeled for use on hemp, but it did work the way we expected. But also the Silmatrix, the Lifeguard alternating with Double Nickel, and then the Double Nickel all reduced disease significantly compared to the untreated control. So that was great. So we decided to, next slide, to repeat this experiment in 2021 with just a few minor changes. So this past year, we used a different cultivar, white CBG, which we had more information to know that white CBG was a more susceptible host to powdery mildew. And then my inoculum was made in the same way as the year before. And but this time I just inoculated 1 time. I rated the same way and collected flowers in the same way as 2020. Next slide, please. So here are our results from 2021. Again, our treatments on the x-axis, AUDPC on the y, and 2 keys letters. And again, our untreated control had the most disease, and our azoxystrobin again worked quite well. And then once again, we saw the Lifeguard alternating with Double Nickel, the Silmatrix and the Double Nickel also significantly reduced disease severity compared to the untreated control. So the second part of this project was looking at cannabinoid production. So there's lots of different cannabinoids that are produced by hemp, and we just looked at a few to get an idea. And we looked at a lot, but I'm going to just talk about a few. We looked at, so today I'm going to talk about total potential THC, CBD, CBG, and CBC. So in this experiment, I collected flowers from each of the treatment plots and looked at the levels of all of those. So next slide, please, Patrick. So when we looked at THC, we saw that there was no effect on the total potential THC. So there's two graphs here, both from 2020 and 2021. The x-axis shows all of the different the treatments for each year, and then the y-axis is showing the total potential THC for each of those treatments. And we didn't see any difference between any of the treatments in either year. Next slide, please. So next, we also looked at total potential CBD. And again, very similar type of graph. Our treatments are on the x-axis and the total potential CBD on the y. And again, there was no differences between the different treatments in either 2020 or 2021. And once again, we saw the same thing with the total potential CBG. Next, please. And again, the same for the total potential CBC. One more. So we also looked at whether powdery mildew infection would affect cannabinoid levels. So for this, I had a separate plot that was both uninoculated and untreated with any powdery mildew. And I collected flowers from plants that were from that untreated control plot. So that's our powdery mildew. Those were inoculated. And then from that separate plot that were uninoculated. And for total potential THC, CBD, CBG, and CBC, I didn't see any significant differences in those cannabinoids between plants that were infected with powdery mildew and plants that were uninoculated. And yeah, so to wrap up, some of the products that are available for growers to use in New York were effective at reducing powdery mildew disease pressure. So yay, good news. And those fungicide treatments and powdery mildew both did not affect cannabinoid production in hemp. And that's all.
Great, thank you, Allie. Gary covered a couple of diseases I was going to highlight because we saw them quite a bit in the last 2 years. I'll cover a few things. My lab is continuing to work in addition to fusarium and powdery mildew on hemp downy mildew. We also in my lab work on cucurbit downy mildew. And one of the questions was how rapidly will it spread? And the picture that you see here is an inflorescence. This was from 2020 in the plots that Savanna, Shelnutt, and the Bergstrom lab were rating. And it actually, it didn't kill the plant. So if this were cucurbit, a cucumber and cucurbit downy mildew, the plant would be dead fairly quickly, maybe 2 weeks. Maybe depending on the weather. And the plants did not die, but they are in no way healthy, right? If you look at this flower, it's not something you're going to want to harvest and sell right away. So I think, as Gary said, we have a lot to learn about some of the diseases that are moving into our hemp crops. This particular pathogen, much like the powdery mildew, will spread via wind. We don't yet know how far, for closely related organisms, we have a better idea as to how far the sporangia might fly in the wind. And we don't yet know for downy mildew, but based on hop and cucurbit downy mildews, we can, assume that it could be a mile. They are the spores. Actually, if you go to the next slide, Patrick, the symptoms Gary showed really nicely. And then in the next slide, you can see the sporangia, they're gray. And so they have some protection from ultraviolet as they're flying in the air, and they don't seem to desiccate really quickly. One of the things that we have seen is that so on the bottom of the leaf in the upper left-hand corner, you see the underside of the leaf with the gray spores being formed. And then in the lower left-hand, you see the spores or sporangia. Well, in water, those sporangia will actually release swimming zoospores. And there's a picture of a zoospore in that slide. And it's those zoospores that will actually germinate and cause the infection on the leaf. In some downy mildews, the sporangia, the spore, the larger spore, can directly germinate if there's not enough water. Hop downy mildew can do that, but in cucurbit downy mildew, you only have the production of zoospores. The reason that's important is because there are different control products that are effective for different types of spores in the pathogen. The other thing that unfortunately we have found is the overwintering spore. So on the right-hand side of the slide, you can see it says within leaf tissue, that thick brown circle, that's called an oospore. It's the overwintering spore, and those will survive in the soil. So if you plant hemp the next season, you are going to have that inoculum already in the soil as you start your season. And these are some of the things that we're thinking about and trying to figure out, okay, what experiments do we really want to look for if we think hemp downy mildew is going to show up more frequently. Next slide. So what are we working on? We are screening as much germplasm as we can for resistance. For some pathogens against this type of oomycete pathogen, in tomato and potato, there's actually quite a bit of resistance that can be found. It's not always used in agriculture, but there are resistance genes there. For cucurbit downy mildew, there's really not as much. In all the screening that's been done, there's really not a lot of resistance available in the plant host germplasm. So we're going to be screening a whole bunch of varieties and seeing where we come out for hemp downy mildew. We also hope to sequence the pathogen genome to get an idea, is it really more closely related to the hemp or the hop? Again, that might give us better clues into control strategies. And one of the reasons I really wanted to highlight this is we are collecting isolates to look at pathogen diversity. So do we all have the same thing? When we see it, we did get an isolate from Massachusetts last year. Unfortunately, it died. One of the things that's really hard working with this pathogen is that you can't grow it in culture. Like Patrick showed cultures of Fusarium, you can't grow this pathogen in culture. It has to be grown on living tissue. So that makes it a big challenge. So as Gary said, we really do recommend that you send samples to the diagnostic lab. They're really excellent and have had a lot of experience now in diagnosing hemp diseases. But if you think you might have downy mildew, we would love to have isolates of the pathogen, and I think the same is true of the Fusarium root rots. I know this is bad for growers, but as researchers, we are looking for the most aggressive pathogens so that we can include those in our tests. So if you do have aggressive, plants that are dying from either root rots or what might be downy mildew, we would love to have those samples. And then Patrick, you can just go to the last slide. The next, yep. I want to make sure that we highlight the Department of Ag and Markets in New York State. They have provided all of the funding for the work that we have done in the lab that we presented today, and they have enabled New York to really get a jump on some of the hemp research compared to other states. And so we really want to thank the state of New York. And with that, Patrick, you can stop sharing and we're happy to answer any questions or move on. Oh, we caught up some time there, Mary. Like 2 minutes.
Yes, you did. Thank you.
Yeah.
I actually have a question. So Patrick mentioned using double nickel as a fusarium control. Is that really like going to work soil applied? Or is it a drench or a spray or a dip for the what? How is that?
Yeah,
gonna
we did a drench on the soil.
And in talking to Certus, they didn't think it would work, but it was one of those,
okay.
well, if you're doing the trial,
Okay.
let's give it
I
a
was
shot.
like,
and
what
see.
does that work on? Does it work on other things? Can we okay?
No.
Because I had only ever heard of it as a foliar applied. All right. So it was like real research on there. That was the only question I had. And I will I do want to mention again that the roots on these on hemp are just so sad compared to other crop. I mean, they're just, Patrick showed those pictures of, fusarium and that the roots didn't really spread out. They're really, if you don't dig the hole correctly for the transplant, even without disease, you can have a problem because they're just not as aggressive as you would expect. That amount of top growth, like a little Christmas tree, you would expect the bottom to have more oomph, and it just doesn't.
The other thing I'll highlight, Mary, is that Allie has done a really nice host range study with powdery mildew, and those results from this past summer will be posted up on the hemp website.
Okay, that Janice posted before. Good.
Yep.
So just to get started, I just want to go into a little background on what a full season plant and what an autoflower plant is. So on the left side here, you have a full season plant, which is day length sensitive. And on the right, you have an autoflower plant, which is also known as a day length insensitive plant. And so our full season cultivars are triggered to flower when the critical night length reaches a certain threshold. So that means that the plant receives a signal to flower as the nights increase in length. And our autoflowers don't receive that signal, so they're flowering independently of day length. And just some general notes, full season plants usually are larger and have a longer time to maturity at around 3 to 4 months. And they're usually planted at a lower density than autoflowers, which are relatively smaller in stature and have a shorter time to maturity at 2 to 3 months. And they're usually planted at a much higher density. And so this short time to maturity can enable double cropping with another round of autoflowers, or you can come in and follow up with something that's going to grow late into the season, like a brassica. So low density versus high density. So when I talk about low density plantings, I'm referring to a traditional horticultural style planting, usually spacings around 4 foot by 6 foot, which results in roughly 2,000 plants per acre. And generally, plants are transplanted into these plantings with a water wheel transplanter or by hand. And they can be planted into bare ground or into horticultural plastic to control weeds. And generally, these plantings are irrigated with drip irrigation. On the right side over here, we have a high density direct seeded planting.
Savanna, this is Janice. Your slides aren't advancing. You're on the full season versus autoflower slide still.
Thank you so much, Janice. I appreciate it. I'm not just so you can see this picture of autoflowers really quickly because I don't think you guys saw it. This on the end, full season plants, left side, we have our full season, right side, we have autoflowers. All right, so just once again, low density versus high density. Over here on the left, we have our low density planting. On the right, high density direct seeded. So spacing usually around 1.5 feet by 2.5 feet. And we have roughly 11,600 plants per acre in this situation. And, generally this is direct seeded with a tractor or push behind seeder, and irrigation happens with a sprinkler or furrow irrigation. Okay, so just a little bit of background on direct seeding. Direct seeding enables mechanized planting, maintenance, and harvest of large acreage, which enables one person to do more per acre on their farm as long as they have the proper equipment. You're able to avoid transplant shock in autoflowers. So autoflowers are really sensitive to any sort of root disturbance, and that can cause them to be stunted. Additionally, you decrease use of plastic. You can't use plastic in this sort of situation. So if you're concerned about sustainability, you're immediately eliminating that in your production system. Some cautions: weed management is very intensive until canopy closure occurs, and within some of the autoflower cultivars, canopy closure will not occur at all. And additionally, establishment can be quite difficult in wet years, and dry years require you to be really on top of your irrigation to get an even stand. So in 2020, in conjunction with Oregon State, we hosted a direct seeded field trial at Bluegrass Lane. And we had 4 full season and 5 autoflower cultivars. And we planted those individuals on June 30th, and we harvested August through September. So you can see just a list of the cultivars we used, and their day length sensitivity is right here in this table. So we direct seeded our plants, our seeds at 1/4 of an inch deep. And we planted 1 seed every 5 inches with the polyplanter junior, which is this push behind seeder that you're seeing right here. We had 30 inches between our rows, and we thinned our autoflowers to 1 plant per foot. And we thinned our full season plants to 1 plant for every 1.5 feet. And we did that thinning once the plants had roughly 3 sets of true leaves. And you can see an unthinned row here and a thinned row right here, down to about thin to 1 plant for every 1.5 feet here. So final percent emergence. So prior to thinning, we counted the number of plants in a row to determine the final percent emergence. This is the percentage of plants that successfully germinated and established themselves. And as you can see, our numbers are relatively low, ranging from around 30 to 50%. So some potential causes of low numbers are seedling fungal diseases, herbivory by insects or animals like rabbits or deer, birds eating seed right after planting improperly cleaned seed, and having an incorrect planting depth. So it's important to keep these losses in mind if you are a producer, because really you saw that we overseeded by like 3 times the amount of seed that we needed in some cases. So this really justifies the need for overseeding by a lot. And this is tough for some producers because the price of seed is so high, they can't really justify spending so much money on seed. So it's just something to keep in mind. It's really necessary to establish a successful stand. Okay, so at flowering time, we rated plant shape to get an idea of the overall plant architecture within a cultivar. And so just really quickly, plant form can be influenced by genetics and environment, and it gives an idea of the level of segregation for within a cultivar. So if a cultivar is segregating heavily for form, perhaps it's also segregating for other traits like leaf color or cannabinoid phenotype, etc. So additionally, some shapes are thought to possibly be better for a mechanized harvest. We're not exactly sure which ones those are yet, but it's thought that columnar or pyramidal shapes feed better onto a stripper header than a candelabra shape. So over here you see our columnar plant. It's very tall and narrow. It has strong apical dominance and limited branching. Our pyramidal shape has a strong central leader and some branching down here on the bottom, and our candelabra has weak apical dominance and extreme side branching. So just as an example over here in this plot, it's highly uniform. You see that for the most part, all the plants are pyramidal. And over here on the right, this plot is segregating for shape. We have a mix of a pyramidal shape and a columnar shape. Okay, so overall, you can see that most cultivars are heavily segregating for form. And white and pipeline seem to be the most uniform. Pipeline's mostly columnar, white is mostly pyramidal. And Auto CBG, Queen, Dream, and Rogue are the least uniform cultivars. So just something to keep in mind when selecting your cultivars. This might give some insight into the overall stability of the cultivar. So after harvest, we dried our plants and stripped off the floral components, and we took their weight. And so this graph is depicting the total yield per acre for each cultivar in pounds, and it is corrected for planting density and seedling emergence. So as you can see, Auto CBD is the lowest yielding at roughly 1,500 pounds per acre. And at the end of the day, when you come down to like, what is the yield for a single plant? It's very low. It's 1/10 of a pound per plant. And a white is the highest at roughly 3,800 pounds per acre. And that comes out still quite low to around 1/3 of a pound per plant. It's important to keep in mind that since these plants are so close together, they're spending a lot of time building stem components, and they're only really producing floral biomass on the very tips where they're getting a lot of light penetrance into the canopy. So an additional note here is that autoflowers are generally lower yielding than full season plants. So yeah, just something to keep in mind. So direct seeding, just a quick cautionary tale. We attempted to recreate this trial this past year, but as some of you may know, it was an extremely wet year, and unfortunately this trial washed out. We were unable to replant. So if you're interested in direct seeding, just be wary of the fact that this could happen to you. All right, moving on into high tunnels. So why might someone choose to grow on a high tunnel? So a high tunnel enables a producer to extend their growing season roughly from May through to October. There's a potential for yield benefit, which we're exploring right now. And there's potential for multiple cycles of hemp in the high tunnel per year, which we also explore in some data I'm going to show you guys right now. And additionally, it just allows producers to diversify their production within their own high tunnels. So in 2021, we conducted a trial as part of a larger New York Farm Viability Institute funded trial to determine if double cropping in high tunnels is a viable strategy. So we planted 3 full season and 3 autoflower cultivars in the high tunnels, and we hosted a high tunnel in Geneva at the experiment station and also in Cortland at Main Street Farms. So thank you, Main Street Farms, for the use of your high tunnel. So we decided to start our plants in rock wool, our compressed soil blocks, to avoid irritating the autoflower's roots. Like I said earlier, they're very susceptible to becoming stunted if anything happens to their roots. And then we planted into a staggered double row design. And we planted our autoflowers at 1 plant per foot this way, and our full season at 1 plant per every 2 feet that same way. And there were roughly 2 feet in between the stagger plants. And as you can see, we just laid down some landscape fabric between rows, and then we used drip irrigation. So after harvest, we stripped and weighed floral biomass. The gray boxes are results for Geneva, the yellow are for Cortland, and the yield was generally higher in Cortland. Autoflower yield ranged from 1 ounce all the way to 2 ounces. And on average, and full season yield ranged from 1 ounce to 5 ounces per plant. So just some takeaways here. It's very difficult to get the timing down so that the full season plants are transplanted well before they reach their critical night length, which is their signal to flower. And so you have to be really on top of your scheduling when it comes to, when you're going to start your transplants that are immediately following your autoflowers, and be sure to get your autoflowers in early enough so that they're mature in time. And it might be useful to be sure to use a very late flowering cultivar in the high tunnel when it comes to the full season plants. And, honestly, I would like to repeat this trial at some point with earlier planting dates, and just knowing what I know now about scheduling transplants, I think we might get some different results. So we had 2 cultivars that were both direct seeded and in the high tunnel. And as you can see, generally yield was much higher in the direct seeded plantings. So this really just emphasizes the importance of not disturbing the autoflower's roots. And it suggests that it might be a good idea to direct seed into the high tunnel with a polyplanter, just to avoid the need for transplanting in the first place. So moving on, this past year, we noticed a lot of powdery mildew in our high tunnels. So as Allie just went over, it's caused by the Ascomycete in the Glavinomycetes family. And it is a persistent problem in low airflow, high humidity environments like high tunnels. And so in 2022, we planned a silicone application trial because this has proven to, because soil-based applications have proven to be really effective in pumpkins and wheat. And so we want to see if this is the case for hemp, and it might enable growers to control their powdery mildew without having to spray. And so with that, I would just really like to thank Janice at Cornell Cooperative Extension for all of her help in the high tunnel, Dr. Jeff Steiner for his help in procuring seeds for the EOH trial, the Vians Lab for their help, Main Street Farms for their high tunnel, our seed donors over here on the right side. And I'd like to thank Oregon State, the Farm Viability Institute, Empire State Development, Agri Markets, and USDA for funding. So with that, I'll take any questions.
Anybody got, oh, I see some clapping from Heather. Yay. I have a question. So on your weights for flower mass, is that wet weight or dry weight?
That's a dry weight.
Okay.
Yeah.
Because that can make a big difference when you're
Yes.
talking about an ounce or two. That could be serious.
Definitely.
And something about, oh, the silicone. So Chris is still on here too. So you spray it on the soil and then the spores can't get up and get on the plant. How does it work?
Yeah, the idea is that you drench the area or incorporate it into the soil, and the plant uptakes it into the leaves, and it inhibits the ability of
Really?
powdery mildew to reproduce on the leaves
Cool.
or establish itself. Yeah.
Yeah, it'll be exciting to see how all that works. A colleague was telling us that that's what they do in the greenhouse, but they have to put more silica in for new growth, right? Yeah, that's what he was saying. So I think it's a cool study that Savanna will be doing.
Yep, we'll see how it goes.
That sounds interesting. Any other questions?
The sill matrix that Allie put on was foliar applied. So it's a different idea.
Right. That's why I wasn't, because then the last slide was something else. And so,
Yeah.
Okay.
Hello, thank you all for coming. I'm Dr. Carlyn Buckler, an associate professor of practice in the School of Integrated Plant Science at Cornell. I lead the Hemp Master Professional Studies program, and I'm so glad to talk to you about this unique opportunity. I started out as a maize geneticist over 30 years ago, and as many of you know, hemp, or cannabis, is not corn or wheat or apples or field greens. Cannabis presents many unique opportunities as well as threats. And for science too, cannabis has been in front of us for thousands of years, but even as the Green Revolution and the advent of genetics and genomics marched on, we have only in the last several years been able to use modern plant science to study this plant that humans have been using for millennium. Cornell University Plant Science has been at the forefront of this research in hemp, and this program allows for a myriad of paths for students to understand this plant and what it can and cannot do, as well as how to be successful in the industry at large. So, what is the Hemp Masters Study? This is a one-year intensive program specifically designed to prepare those who are interested in the production, processing, and/or business of hemp to have a broad-based experience of the industry, as well as a way to become competent in a specific area of the industry. Courses and projects cover everything from genetics, genomics, production, processing, disease and pest issues, controlled environments, and warehouse agriculture. Whether you're interested in growing for flower, herd, or best, specific aspects of the industry, or maybe someday you want to be your own CEO or COO in your own hemp business. Either way, we've got you covered from seed to sales. So let's look at some of the faculty who are involved in the MPS program. Dr. Larry Smart has been leading Cornell's hemp research and extension team and has initiated a long-term breeding and genomic program to develop new hemp cultivars for New York State. Dr. Smart also teaches courses on plant genetics and genomics. Dr. Chris Smart focuses on the management of disease and host pathogen interactions in hemp. Dr. Neil Matson specializes in greenhouse and controlled environment agriculture. Tony DiTommaso studies sustainable agriculture as well as soil and pest management. And Heather Grab teaches the production and processing courses. And let's look into those. So those are seminal to the master's program. So Dr. Grab teaches the hemp production systems, and this course will provide students with knowledge of different hemp production systems for fiber, grain, and cannabinoids. Students will gain an understanding of field, controlled environment, greenhouse, and high tunnel production systems. Topics covered will include hemp physiology, agronomy, cultivation, seed science, nutrition and fertility, harvest methods, and post-harvest storage. While this course cannot provide students with a thorough understanding of all of the intricacies of hemp production, it is intended to provide a survey of approaches and techniques that are used in the production of hemp grain, fiber, and cannabinoids. And the hemp processing course covers the principles and best practices to create a diverse array of food, extracts, and fiber products from hemp. Lecturers will describe key products and production practices, highlight how hemp products compare to those produced from other materials, and discussion sections will help review scientific studies relevant to industrial hemp products and processing, and provide opportunities to interact with key industry leaders. Lab sections will give participants hands-on experience using a broad array of analytical and processing equipment. Students are giving flexibility to tailor some course components towards their own interests as well. This is a plant science overview course that I teach, Cannabis, Biology, Society, and Industry, and the MPS students take this in their first semester. Lecturers from Cornell and from around the US come to the class and talk about their path to where they are now and their expertise from propagation to product, but also about law, policy, business best practices, diversity, equity, inclusion issues, state and federal regulations, pharmacology, veterinary medicine, yes, that's a thing, and much, much more, all of which allows students to explore every aspect of the industry. And this is one of the reasons this MPS program is so good. Yes, the plant science here is world-class, but Cornell is also home to the Johnson School of Business, one of the best business schools in the country, as well as the Dyson School of Economics and Management, the Cornell Law School, and many others, where you can take classes as part of your program. Many of the speakers who come to talk in my class are not only some of the most successful people in the business, but are also graduates from Cornell. So each student in the Hemp MPS program does a capstone project for their Master's of Professional Studies that reflects their goals and interests in the hemp field. Using your mentors and other faculty and staff, students will write a thesis describing the project in full. So here's a couple of examples of what other graduates from the Hemp MPS program have done. One student came to the program wanting to learn hemp farming so they could use their family farm in upstate New York for hemp production. But after being introduced to folks at Cornell Cooperative Extension, they decided to help with a survey CCE was doing to find out what kinds of resources farmers want to facilitate their knowledge and needs for farming hemp. The student did a wonderful job working with CCE on this development and learned much about the understanding of obstacles that farmers of hemp face. Another student looked at the misinformation regarding the value proposition for some farming of hemp. They looked at many of the standard assumptions of production and researched the viability of various outcomes, finding that some were not quite so viable. And another looked at cold tolerance and in specific cultivars of hemp that may help mitigate some ramifications of climate change. But the bottom line really is that the choice is yours in this program to delve deep into what you are interested in for your final project. So again, really happy to talk to you about this. Please come join us. If you have any questions, please don't hesitate to email me. And again, I thank you very much for your time.
Mary, I can also chime in with, we will be soon this month putting out a job advertisement to hire someone to put much of this content online as certificate programs. So that will be enhancing our educational program as well.
Yeah, actually, yeah, I do. I have a question. I've been asked by people whether these online courses are available for everyone, and if so, what are the prerequisites and requirements for them to take these courses?
My understanding is that the only course that's currently online is the hemp breeding and genetics. The other courses aren't, we haven't even hired the person that will be working to put them online yet. So that's in the very infancy. For the hemp breeding and genetics, I don't know. They are open to everyone, but there is a charge. It's not free access.
There's a financial charge.
Yep.
How about prerequisites?
To my knowledge, there are none. But again, I haven't taken any, and I think, yeah, I don't know when we develop the new courses, if, you know, in order to do the 5th one, will you need the information in the 1st, 2nd, and 3rd or not? Since we haven't built them yet, it's too early to say.
Any other questions from anybody?
I will just add a plug-in for the growers and industry members who are in the audience today, that we are always really excited about partnering with people from the industry to help students to develop their capstone projects. So feel free to reach out if that's of interest.
This has been a production of Cornell University, on the web at cornell.edu.